All our genetic information is encoded in DNA. DNA is found in the nucleus of every cell of our body as part of these structures called chromosomes. The analysis of these chromosomes is called a karyotype. A karyotype is performed on cells called lymphocytes, obtained from a simple blood sample, but it can also be performed on amniotic fluid cells obtained during pregnancy. Each chromosome can be distinguished from the rest by its shape, size, and banding pattern, which can be observed along the entire chromosome after specific staining completed in the laboratory. The alterations detected in a karyotype can be classified into two categories: numerical alterations and structural changes. The number of chromosomes is characteristic of each species. Human beings have 46 chromosomes in 23 pairs in the nucleus of every cell in our bodies. These 23 pairs are arranged in 22 pairs called autosomes and a pair of 6 chromosomes, X and Y, which differentiates with the sexes. Women have 2 X chromosomes, whereas men have 1 X and 1 Y chromosome. The most widely known numerical alteration is Down syndrome. In the karyotypes of these patients, there are 3 chromosome 21 instead of the usual 2, causing varying degrees of learning disabilities. There are other numerical alterations that generate reproductive problems. For example, men with Klinefelter syndrome have 2 X chromosomes and 1 Y. These patients suffer testicular failure, resulting in fertility problems. On the other hand, women who suffer from Turner syndrome have only 1 X chromosome karyotype. The absence of 1 X chromosome causes underdeveloped sexual characteristics and consequently leads to infertility. Certain chromosomal regions are found to be duplicated or even absent in structural alterations. This gain or loss of genetic material has extremely varied consequences depending on the genes involved. Other structural abnormalities are translocations and inversions. In the first case, there is an exchange of material between 2 chromosomes. And in the second, a chromosomal region is inverted with respect to its normal position. Structural alterations may have different clinical consequences, such as low semen quality in men or repeat miscarriage in women. If a chromosomal abnormality is detected in one member of the couple, the solution to ensure healthy biological descent is pre-implantation genetic diagnosis, or PGD. PGD is a technique for detecting chromosomal alterations in an embryo pair prior to its transfer to the mother's uterus. In addition to the alterations already mentioned, there are chromosomal variants called polymorphisms. These polymorphisms are relatively common in the general population and show no apparent phenotypic consequences, but recent scientific studies have linked them to infertility. A karyotype is an important diagnostic test in patients who require assisted reproduction techniques because there is a higher incidence of abnormal karyotype in couples with fertility problems. This technique is indicated mainly in cases of seminal alteration, ovarian failure, and in couples with repeat miscarriage or implantation failures.